Namaskar children, good morning. So I hope you read the textbook. I hope you are clear with the part which we learned in the last class. So what did we learn, children? We started with cell organelles, right? So now let us start with the next cell organelle. And what was the cell organelle which we studied in the last class? It was about the endoplasmic reticulum. Now what is endoplasmic reticulum? It is a large network of membrane bound tubes and sheaths. Okay, and it looks like long tubules or round or oblong bags. How do they look like? It looks like either long tubules or round or oblong bags, that is vesicles, right? Vesicles are nothing but rounded structures. Okay, and the endoplasmic membrane is similar in structure to the plasma membrane. So how is the plasma membrane made up of? It is made up of lipid and protein. Same way, endoplasmic reticulum's outer membrane is very similar to the plasma membrane. And what are two types of endoplasmic reticulum? Rough and the smooth endoplasmic reticulum. Right? Now if you take the case of rough endoplasmic reticulum, as the name says, when we view this under the microscope, it has particles called ribosomes attached to its surface. That is why it appears rough. And the ribosomes which are present in the all active cells are the site of protein manufacture. Remember this, ribosomes help in the manufacture of protein. And the manufactured protein is then sent to various parts of the cell depending on the need using the endoplasmic reticulum. And what is the work of smooth endoplasmic reticulum? They help in the manufacture of flat globules or lipids, and it is important for cell function. And some of these proteins and lipids will help in building the cell membrane. So the protein from rough endoplasmic reticulum and lipids from smooth endoplasmic reticulum will help in building the cell membrane, and that process is known as membrane biogenesis. And some other proteins and lipids function as enzymes and hormones. And what are the functions of endoplasmic reticulum? Their one main function is to serve as a channel for transport of materials. Okay, and then night our work in materials transport cheyanana, they are acting as a channel, pathway. Okay, and especially which material? It is the protein. Okay, and where do they transport it to? They transport it between various regions of the cytoplasm or between the cytoplasm and the nucleus. On the cytoplasm le thannulla pala organelles vendi organelles lokke proteins chodukan channel aayittu act cheyum, or else they will be moving between the cytoplasm and the nucleus, right? And the endoplasmic reticulum also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. Other than that, other than producing, transporting this protein, they also act as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. Cellil നടക്കുന്ന ചില biochemical reactions നടക്കാൻ അവർ place ആയിട്ട് act ചെയ്യുന്നുണ്ട് in the cytoplasm, right? And in the liver cell of group of animals called vertebrates, the smooth endoplasmic reticulum plays a very crucial role in detoxifying many poisons and drugs. Okay, for smooth endoplasmic reticulum, vertebrates in vertebral column organisms like human beings, many animals, in them they play a very crucial role. And what role do they play? They help in detoxifying many poisons and drugs. അവരുടെ ശരീരത്തിലേക്ക് പോകുന്ന പലതരത്തിലുള്ള പോയ്സൺസും അതുപോലെ തന്നെ ഡ്രഗ്സും, chemical drugsum, it is being detoxified. Detoxified means it is to remove the toxin. Okay, toxin എന്ന് പറഞ്ഞാൽ അപകടം വരുത്തുന്ന സബ്സ്റ്റൻസസിനെ റിമൂവ് ചെയ്യാനും ഇവരെ സഹായിക്കുന്നുണ്ട്, right? So now let us move on to the next cell organelle, which is called as Golgi Apparatus. So now let us learn about our today's topic, children, that is Golgi Apparatus. What is it? Golgi Apparatus, right? So this Golgi Apparatus was first described by Camillo Golgi. So his name has Golgi, so they gave a name to it which is called as Golgi Apparatus. So he was the one who first described the Golgi Apparatus, right? And how is this Golgi Apparatus made up of? It consists of a system of membrane bound vesicles, right? And how are these vesicles? They are flattened sacs. vesicles എന്ന് പറയുന്നത് normally these are empty spaced structures, right? So it can be round or it can be flattened like Golgi apparatus, right? So this Golgi apparatus, it consists of a system of membrane bound vesicles. What is this membrane? You know that membrane is the outermost covering of a organelle. Like a cell, it also, organelles also has membrane. So it is a system of membrane bound vesicles. Okay, so they have an outer covering and they are bound vesicles. It means that they are attached one to one. Okay, and how are these vesicles? They will have flattened vesicles. You can see many vesicles here, right? So in between you can see an empty space. അതിനോണ്ടാണ് അതിനെ vesicles എന്ന് പറയുന്നത്, right? And these are flattened sacs. Okay, sacs എന്ന് പറയുന്നത് structures, I mean empty structures നെയാണ് നമ്മൾ flattened sacs എന്ന് പറയുന്നത്. And how are they arranged? They are arranged approximately parallel to one another. What is this parallel? It is like this one, parallel to another. You can see it here, right? So they are seen parallel to one another. Okay, clear with this, children? So how is the Golgi apparatus? So Golgi apparatus, it consists of a system of membrane bound vesicles. What does it contain? It contains a system of membrane bound vesicles. You can see many of the vesicles which are arranged parallelly to each other in stacks, which is called as cisterns. Okay, so when these vesicles are arranged parallelly to each other, we are calling those stacks as cisterns. ഈ സ്റ്റാക്സിനെ നമ്മൾ cisterns, stacks എന്ന് പറഞ്ഞാൽ ഒന്നിനെ അടുപ്പിച്ച് ഒന്ന്. Okay, so these stacks are together called as cistern. What is it? Cisterns, right? Single one is cisterning and together it is called as cisterns. You can see it here, it is written as cisterns. Okay, now along with this, the connection, the main work of this Golgi apparatus, so their work is to collect materials from endoplasmic reticulum. So what happens? Their membrane, so these membranes will often have, whose membrane? The Golgi apparatus membrane will often have connections with the membranes of endoplasmic reticulum. You can just see here, I will just show it here in this image. Can you see here, children? Can you see the endoplasmic reticulum here? And next to the endoplasmic reticulum you see this Golgi apparatus, right? So these membranes often have connections with the membrane of endoplasmic reticulum. So they will have some connection with this endoplasmic reticulum. Okay, and therefore constitute another portion of a complex cellular membrane system. Okay, so and it gives rise to a complex cellular membrane system. Okay, so these membranes often have connections with the membranes of endoplasmic reticulum and therefore they constitute another portion of a complex cellular membrane system. ഒരു cellular membrane system ന്റെ വേറൊരു complex portion ആയിട്ട് ഈ Golgi apparatus മാറുന്നുണ്ട്. Because they are connected to one another. They are connected with the endoplasmic reticulum. Right? Now, what happens here, as I told, as they are connected with each other, that is endoplasmic reticulum and Golgi apparatus, when as they are connected to each other, the material synthesized, what are the materials synthesized in endoplasmic reticulum? Yes, it is protein, and we saw that smooth endoplasmic reticulum will produce lipids. Okay, so other than that, some of these proteins will act as enzymes and hormones. That is, proteins and lipids will act as enzymes and hormones. So the materials which are synthesized near the endoplasmic reticulum is packed and dispatched to various targets inside and out of the cell, outside of the cell through the Golgi apparatus. So what is the function of this Golgi apparatus? So suppose if this is endoplasmic reticulum, so think this is an endoplasmic reticulum and it is connected to the Golgi apparatus. Think that it is connected to the Golgi apparatus, right? So what they do, the substances which are synthesized in the endoplasmic reticulum will move to the Golgi apparatus. Okay, so endoplasmic reticulum will transfer their produced or synthesized substances into the Golgi apparatus. Okay, and inside the Golgi apparatus they will be stored. ഈ സിസ്റ്റേൺസിൽ ഈ ഓരോരോ സ്റ്റാക്സിലും അവരെന്ത് ചെയ്യും, ഈ മെറ്റീരിയൽസിനെ സ്റ്റോർ ചെയ്ത് വെക്കും. Okay, and whenever a cell needs it, it will pack the material and it will dispatch it to various targets. അപ്പോൾ ഇവിടെയുള്ള സബ്സ്റ്റൻസസിന് ആവശ്യം വരുന്ന സമയത്ത്, സെൽസിന് ആവശ്യം വരുന്ന സമയത്ത് they will pack inside these vesicles, rounded vesicles. Okay, can you see the new formation of vesicle here? So from the cisterns there will be formation of such rounded vesicles, and inside the rounded vesicle they will pack these substances and they will dispatch it to various targets. Dispatch എന്ന് പറഞ്ഞാൽ എന്താ, അവിടുന്ന് they will make it move to the target place, and this target can be either inside of the cell and it can be outside of the cell. Okay, and how is it done? It is done with the help of this Golgi apparatus. So the material synthesized near endoplasmic reticulum is packed and dispatched. Okay, so what does the Golgi apparatus work? It has to pack and dispatch. Pack either dispatch ചെയ്യണമെങ്കിൽ what they have to do first, they have to receive the materials from endoplasmic reticulum. Okay, so they will receive the materials from endoplasmic reticulum, they will store in their cisterns or stacks, and whenever needed they pack these substances in vesicles like this, rounded vesicles, and then they will dispatch it to the various targets inside and outside of the cell through the Golgi apparatus. So this is what Golgi apparatus do. Okay, അവർ സ്വന്തമായിട്ട് സബ്സ്റ്റൻസസ് പ്രൊഡ്യൂസ് ചെയ്യുന്നില്ല, ഇൻസ്റ്റെഡ് they will help in packing and dispatching the substances or materials from the endoplasmic reticulum. Clear, children? Okay, let us learn about the functions of this. ഇപ്പോൾ ഇവരുടെ functions എന്തെന്തായിരിക്കും? You know that they are receiving materials from endoplasmic reticulum, so their function is to storage. That means that they help in storing the substances. Miss പറഞ്ഞിട്ടില്ലേ, Golgi apparatusൽ they will receive the substances from endoplasmic reticulum and they will store it in the Golgi apparatus. Okay, and modification can also be done. എങ്ങനെ മോഡിഫിക്കേഷൻ നടക്കുക? എൻഡോപ്ലാസ്മിക് റെറ്റിക്കുലം പ്രൊഡ്യൂസ് ചെയ്യുന്ന സ്മോൾ സിമ്പിൾ പ്രോട്ടീൻ സബ്സ്റ്റൻസസ് ക്യാൻ ബി കൺവേർട്ടഡ് ഇൻ ടു കോംപ്ലെക്സ് വൺ ബൈ ദീസ് Golgi apparatus. എൻഡോപ്ലാസ്മിക് റെറ്റിക്കുലം തരുന്ന സബ്സ്റ്റൻസസിൽ മാറ്റങ്ങൾ വരുത്തുക. മോഡിഫിക്കേഷൻ മീൻസ് ഇറ്റ് ഇസ് ടു മേക്ക് ചേഞ്ച് ഇൻ ദ സബ്സ്റ്റൻസസ്. Okay, so their function is to help in storage, modification and packing of products in vesicles. We saw the small vesicles, rounded vesicles forming, right? So they will pack their products in the vesicles. And in some cases, complex sugars may be made from simple sugars in the Golgi apparatus. So inside the Golgi apparatus, they also help in making complex sugars from simple sugars. And this is not happening in all the cells, it happens in only some cases. And along with that, another main function of Golgi apparatus is to help in the formation of lysosomes. So this is a very, very important cell organelle which we will learn in the next part. So what is it called? It is called as lysosomes. What is it? Lysosomes. Right? Now see here, children, you can see the Golgi apparatus. You can see the stacks which is arranged, right? So these are membrane bound vesicles. You can see the spaces in them. So these are membrane bound vesicles which are arranged parallelly, each after the other, and they are like stacks which are called as the cisterns. Right, and they will help in packing and dispatching of substances which they receive from endoplasmic reticulum. And other than that, they will store these substances, they can bring about modifications, and they do packing of products in these vesicles. Right, ഈ secretory vesicles എന്ന് പറയുന്ന rounded structures ലായിരിക്കും modified structures ഉണ്ടാവുക, or the substances will be there. Right, and this will be transported either inside within the cell or outside the cell. സെല്ലിൻ്റെ ഉള്ളിൽ നിന്നും അതുപോലെ തന്നെ സെല്ലിൻ്റെ ഉള്ളിൽ ബാക്കി വേണ്ടിയ സ്ഥലത്തേക്കും, or else it can move even out of the cell. Okay, and other than that, in some cases, complex sugars may be made from simple sugars in the Golgi apparatus, and they have another main function, that is they are involved in the formation of lysosomes. Don't forget it. They are involved in the formation of lysosomes. Clear with it, children? Okay. Now let us move on to the next cell organelle, which is the lysosome. Now let us learn about lysosomes. What is it? Lysosomes. Let us see structurally how are these lysosomes. First let us learn. So structurally, lysosomes are membrane bound sacs filled with digestive enzymes. So they are membrane bound. You can see the outer membrane here, right? So they have marked it as membrane. So these are membrane bound. So this is the complete lysosome. Adinnoru half murichittana kanikkunnathu. Okay, you can see the membrane, and these membrane bound sacs which are filled with digestive enzymes. Sacs എന്ന് പറയുന്നത് empty space ഉള്ള സ്ഥലമാണ്. Okay, and ആ empty spaces എന്താ ഉള്ളത്? They are filled with digestive enzymes. Right? So structurally, lysosomes are membrane bound sacs which are filled with digestive enzymes. Okay, and where are these enzymes made? These enzymes are made in the rough endoplasmic reticulum. Okay, hope you remember about what we learned in endoplasmic reticulum. We told that some of the proteins will act as enzymes also. Okay, so these enzymes are made by rough endoplasmic reticulum. Okay, now what is the work of this lysosomes inside a cell? Lysosomes are a kind of waste disposal system of the cell. ഈ ലൈസോസോം എന്ന് പറയുന്നത് എന്താ? ഒരു വേസ്റ്റ് ഡിസ്പോസൽ സിസ്റ്റം ആയിട്ടാണ് ആക്ട് ചെയ്യുന്നത്. ഓക്കെ, ഒരു സെല്ലിൻ്റെ വേസ്റ്റ് ഡിസ്പോസൽ സിസ്റ്റം എന്ന് പറയുമ്പോൾ അതിന് ആവശ്യമില്ലാത്ത സബ്സ്റ്റൻസസ് വന്നാൽ അതിനെ ഡിസ്പോസ് ചെയ്യാൻ ഹെൽപ്പ് ചെയ്യുന്ന ഒരു ഓർഗനലാണ് ലൈസോസോം. So lysosomes are a kind of waste disposal of the cell. Right? So these help to keep the cell clean by digesting any foreign material as well as own out cell organelles. Okay, as they have this hydrolytic enzymes. Hydrolytic enzymes പറഞ്ഞാൽ enzymes who have capacity to destroy substances. Right? So this will help them to keep the cell clean by digesting. ഇങ്ങനെയാണ് ആ സെല്ല് മൊത്തമായിട്ട് ഒരു സെല്ല് മൊത്തമായിട്ട് അതിൽ വരുന്ന unwanted substancesന് they will clean by digesting it, or any foreign particle which enters it is also getting digested by the lysosomes as well as the worn out cell organelles. Worn out cell organelles എന്ന് വെച്ചാൽ സെല്ലിൻ്റെ ഉള്ളിൽ ചില organelles there are chances for them to get ruptured or damaged. Okay, അപ്പോൾ worn out എന്ന് പറഞ്ഞാൽ it is to what to say, the damaged cell organs. ചില cell organelles സെല്ലിൻ്റെ ഉള്ളിൽ damaged ആയിരിക്കും. Okay, and damaged cell organ is not good for the cell. So these lysosomes will help in digesting, and they will remove these all substances like foreign materials and worn out cell organelle. Foreign materials എന്ന് പറയുന്നത് it can be some infection causing microorganisms. Okay, so they keep the cell clean by digesting all these unwanted substances. Okay, so now let us see the functions of lysosomes. So let us see how the foreign materials are destroyed. So foreign materials which is entering the cell, such as the bacteria or food as well as old organelle, will end up in the lysosome. ഓക്കെ, ഈ ഫോറിൻ മെറ്റീരിയൽസ് ആയിട്ട് വരുന്നതെന്ന് വെച്ചാൽ ആ സെല്ലിന് വേണ്ടാണ്ട് ആവശ്യമില്ലാത്ത ഏതെങ്കിലും മെറ്റീരിയൽസ് അതിൻ്റെ ഉള്ളിലേക്ക് വന്നാൽ, സോ സപ്പോസ് ഇഫ് ദർ ഇസ് എൻട്രി ഓഫ് എനി ബാക്ടീരിയ, okay, or else any food substance, right? So food substance are nothing but substances like protein, that is peptide എന്ന് പറയുന്ന proteins ആണ്. See, usually our blood will carry digested substances, right? Sometimes these might come as complex substances also. It might enter into the cell as complex substances. അപ്പോൾ അങ്ങനെ ഫുഡ് വന്നാലോ, or else if a foreign particle called as the bacteria, if it enters,